Hello everybody, welcome back to our channel. Today we talk about cell membrane. No, we talked about it before, okay? So we talk about membrane transport, okay? How cell membrane works in transport, okay? So first we have to remember, besides that the lipid bilayers is the basis of a cell membrane. So you remember for lipid bilayers, okay? Outside is a hydrophilic, inside is hydrophobic, okay? And the hydrophilic one attract each other, and the hydrophobic one also attract each other, okay? So the structure is stable, and also the hydrophilic hydrophobic hydrogen carbon there is flexible. So when it in the aqueous environment outside, and then it is for the hydrophilic head, okay? And what is the usage of the cell membrane? Basically, it's a barrier of the cell. Okay? Cell membrane has two layers of phospholipids. Okay? This hydrophobic hydrogen carbon chain inside has no permeability to large molecules and hydrophilic particles. Okay? They don't like hydrophilic string. Okay? So ions and polar molecules can't pass through the middle hydrocarbon chain. Okay? So the cell membrane function as effective barrier between aqueous solution. Okay? So cell membrane separate aqueous environment in cells. Okay? So how can thing pass through the cell membrane? First is by simple diffusion. Okay? You know, hydrophilic one can't pass through. Okay? Iron, polar thing can't pass through, but the small molecule can pass through, especially those are hydrophobic. By simple diffusion, the molecule move from high concentration region to low concentration region. Okay? Just like some probability thing, right? Yeah, high concentration connected to low concentration. Okay, the thing will move from high to low, right? Due to probability. Okay? Low energy or ATP is needed. So we call it passive transport. Okay? Cell membrane is partially permeable. Small molecule can diffuse through it. Okay? As the membrane has a hydrophobic bilayer inside. Okay, so hydrophilic molecule cannot pass through. So water can't pass through it. Okay? Water can't go through the cell membrane by osmosis. Osmosis is by aquaporin. Okay? Only hydrophobic molecule can diffuse through a cell membrane. So osmosis, the simple diffusion of hydrophilic water molecule does not happen through the cell membrane. It's through the aquaporin in the cell membrane. Oxygen, carbon dioxide, and lipid are examples that can pass through the cell membrane via simple diffusion. Okay? And there is a diversified membrane protein. You need to know the names, the meaning for them, okay, and not. Okay? They can be the channel for passive transport, okay, facilitated diffusion, also from high concentration to low concentration, okay. But they are especially for those this is hydrophobic, so maybe a special hydrophilic channel for them, okay. And pump for active transport against the gradient, okay, against the concentric gradient from the low potential, low concentration one to high concentration. Cell adhesion, cell recognition, and cell-cell communication. Binding site for homos, receptor for neurotransmitter electron carriers, and enzyme immobilization. We'll talk about the first few first, okay? And you need to remember all the things. Very easy to remember all the protein. First of all, remember the names, okay? Peripheral protein on the surface, of course. Glycoprotein on the surface, how to distinguish them. Glycoprotein has a carbohydrate chain, okay? Cholesterol is partially inside, and then integral is also partially integrated, okay? And some integral protein is completely going through it. We call it a transmembrane protein. If the transmembrane protein has a channel inside it, we call it a channel protein, okay? And the pore is outside, okay? So very easy to remember, hydrophilic outside. So all the things touching this part should be hydrophilic, okay? The protein, hydrophilic. Inside is hydrophobic. So the protein there should be hydrophobic. For example, cholesterol. Cholesterol is the special, is for the enhance the structure of the cell membrane. Okay. Hydrophilic molecules attract each other. Hydrophobic molecules also attract each other. Very fun. Yeah. The surface of the cell membrane is hydrophilic, so hydrophilic protein amino acids are there. Okay. Inside is hydrophobic. Okay. The core of the phosphorylated polymer is hydrophobic. Okay, so hydrophobic protein amino acids are inside. Okay, inside hydrophobic. Okay, and some are amphithetic. Okay, for example, cholesterol amphithetic. Okay, it has two parts. Okay, so it can be partially inside. Okay, and integral protein have parts of them embedded in the bilayer. They have a hydrophobic middle region and a hydrophilic hand. Hydrophobic. I've told you. It's the way to remember them. Where are they? On the surface must be hydrophilic. Inside must be hydrophobic. Okay. A transmembrane protein is an integral protein that goes through it. Okay. And the channel protein is like a transmembrane protein, but they have a pore channel inside. Okay. And then we want to learn about osmosis. Osmosis is water moving from a high concentration of water to low concentration of water. But we don't call about concentration of water, right? So we call about solid, okay? Solid concentration. So water osmosis from low concentration of solid to high concentration of solid. Yeah, I do remember. So remember, it's concentration of the water or the concentration of the solid, okay? So remember, low solid concentration is high water content. Okay, high solid concentration is the low water content. Okay, so water osmosis from high water content to low water content, right? So from a low solid concentration to high solid concentration. So sometimes it's quite confusing. So osmosis is diffusion of water from low solid concentration to high solid concentration, okay, through the partially permeable membrane. And in the cell membrane, it doesn't allow water to pass through. Okay, so it need to have pass through the aquaporant. Okay, osmolarity. Okay, osmolarity is a measurement of the concentration of the solid. Okay, high concentration of solid is a high osmolarity. Okay, and we call it hypertonic. Low osmolarity, low solid content, we call it hypotonic. Same osmolarity is isotonic. Okay. Water diffuses from a low osmolarity region to high osmolarity region. Remember, water content is reversed. Okay. So when you do the water bathing, you need to be careful. Okay. Make sure low osmosis of water. So the solution and the outside and inside, okay, has to be isotonic. Okay, to make sure low osmosis. For example, when water bathing of the cell, okay, in a hypotonic or hypertonic solution, it will be completely different. For example, if it is isotonic, no problem, in and out the same. If the solution, if bathing in the solution, there is a hypertonic, okay, more solid, okay, also water drinks, okay, and the cell will shrink and facet, okay. When it is in hypertonic solution, bathing, okay, and then water will move in, and then the cell will swallow. If it is animal cell, it will burst, okay. And for the living thing, okay, for example, paramecium, it has a way to control the water content, okay. It has a contractile vacuole to store and then expel the excess water absorbed, prevent the cell from being burst, killed by the hypertonic solution outside. And we even have a meter called osmometer to test the osmolarity of the solution. Okay, we have the solution here, the meter here. At first, this is at the 0 scale. Okay, and then when it is put in the water, okay, pure water, we'll observe whether water will flow inside or out. Okay, if water flow inside or not, it means that it is osmolarity is high. Okay, and then the water level will rise quickly. Okay, so high rises of water level, the initial solution has a higher osmolarity. Okay, got it? Well done. So we talk about facilitated diffusion. You know, diffusion has to be simple diffusion. Okay, has to be hydrophobic. Okay, so we need to have a special channel for the hydrophilic particle to go through, for example, water. Okay, some polar molecule. So it is integral protein going through it. There's a pore inside a specific size for specific molecules. Only specific molecule can pass through it. Still from high concentration to low concentration. Still diffusion. Okay. Still a low energy is required. So we call it a facilitated diffusion. So both the facilitated diffusion and the simple diffusion does not give energy. That's all the energy means that we don't need to provide energy to it, no ATP or no mitochondria, so we call it passive transport. Okay. And what's active transport? Active transport is energy. Active transport is a move from a low concentration region to high concentration region. Sorry, I mixed it up. It should be we was against the concentration gradient from low to high. Impossible, not natural. So we need to give it energy. But when energy comes from, you learn later is from the ATP. ATP comes ATP, okay, energy will come out. Okay. If ATP is inhibited, low ATP, then active transport will be stopped completely. Okay. And the pump protein doing this thing. Okay. Pump protein using ATP to transfer a specific particle up the gradient. Okay. For example, it is a potassium. It is a diagram for it. Okay. It is okay. Outside is a low concentration. Inside is a high concentration. Okay. So the molecule go inside. Okay. And then the channel swapped. Okay. When ATP is used, and then the molecule moved there. So effectively, the solid move from a low concentration area to a high concentration. It's just not the intestine. Okay. How the intestine to absorb the glucose. Okay. Although inside the glucose level is much higher than outside. Okay. So we have the two terms: intracellular concentration, extracellular concentration. You don't know which one is intra and extra, right? So be careful. Okay. So when the intracellular concentration is higher, so laterally diffusion will from inside to outside, right? Passive transport. If you want to reverse it from outside to inside, then you need to active transport. So just think about whether it will happen naturally or not. Naturally is passive transport, no energy is needed. If it is not natural, naturally they don't flow in this way, okay? From low concentration to high concentration, impossible. So we need to ATP, okay? And this kind of channel protein too. Remember the diffuses in and out, diffuses, passive transport, pumped, no matter in and out, active transport, okay? And selectivity in the membrane permeability. Selective means that they just allow some some of the molecule to pass through, okay? The simple diffusion, anything can pass through, okay? So selective maybe it is facilitated one, and also for the active transport one, they are selective, okay? Aquapor one, right? And structure and function of a glycoprotein and glycolipid, it is a extra thing, okay? Not about transport, but it's about membrane protein, so we talk about it also here. Glycoprotein and glycolipids are protein and lipids on the surface of the cell membrane, okay, and they have a carbohydrate there, okay. And the chain of carbohydrate has some binding sites, so it can be used to communicate with other cells, okay. So you can do cell recognition. You can use to detect whether the cell surrounding it nearby is a familiar, friend, or foreign. If foreign, then attack it, okay. It's real. It's how the body has a defense system, okay. Oh, thank you for going through me, okay. All the thing we did, okay. Today talk about transport, right? Cell membrane. Important terms, okay. Passive transport, simple diffusion, facilitated diffusion, active transport, and later on we'll learn more about the active transport. Okay. If you like our channel, remember please help us, okay. Share and like and subscribe. Okay, we are a small channel, we need your help. Okay. See you for next week. Okay. Bye.